So let's talk about the eukaryotic transcription that's done by different RNA polymerases besides polymerase number 2. So there are polymerase 1 and 3 that are transcribing different RNA molecules like ribosomal RNA and transfer RNA molecules, right? So let's talk about them. So in general, some general transcription factors like TARA binding proteins or factor 2F, factor 2E, although they're not exactly the same thing, kind of counterparts, similar proteins are involved in both polymerase 1 and 2 transcription initiation mechanism. Okay, and then polymerase number 1, okay, is dedicated for expression of a single gene, okay, one big gene that transcribes the ribosomal RNA precursor. Okay, from this single precursor, rest of the ribosomal RNA are transcribed together. They are made together. Okay, so that precursor gene that exists as in multiple copies. What does that mean? Multiple copies. There are a number of these gene copies existing throughout the eukaryotic genome. Okay? It's not just a one gene or two genes. There are many of those genes because ribosomal RNA molecules are needed a lot within your cell, right? So that's why you need multiple copies of those. And those genes are always turned on as housekeeping genes, okay? So the promoter for ribosomal RNA gene has core promoter and upstream control elements. Core promoter, upstream control elements, okay? And then the upstream binding protein binds on the upstream core promoter, okay, and then that they recruit SL1 protein, which binds on the TARA binding and transcription factor in the core promoter area. So that's how polymerase 1 transcription is studied, okay. And then polymerase number 3, which transcribes a lot of transfer RNA, okay. Their promoter is located kind of downstream from the transcription start site. So that's a transcription start site, kind of downstream, okay. Especially they are named as boxes, box A and B or C as well, okay. So factor 3B, because they are involved in the polymerase number 3, 3B and 3C for tRNA gene, and the 3A gene for 5S ribosomal RNA are involved, 3 transcription factors, okay? And then 3C binds the promoter region, covers box A and B both, okay? When it binds, it recruits factor 3B, okay? 3C first and the 3B after that, and then that recruits the polymerase number 3, okay? So polymerase number 3 displaces factor 3C, okay, and then it moves along the DNA after that. Okay, so factor B, C, D are important in the polymerase number 3 transcriptional initiation. That's what it says. Okay, and then eukaryotic genes are mosaic, meaning there are a lot of introns and exons. Okay, so consisting a lot of non-coding regions called introns, and the coding regions that are called exons. Okay, so when they are transcribed, of course they are transcribed together, intron exons all together, but right after transcription, those introns should be removed from your transcript. So we call that process as intron removal or splicing. So the process of intron removal from the pre-mRNA is called RNA splicing. Okay, so some pre-mRNA can be spliced multiple ways through alternative splicing. That's a very interesting mechanism because it allows eukaryotic cells to make maybe a lot more proteins than the number of genes, because about 90% of the human genes are spliced alternatively. It's not one gene per one mRNA per one protein. It's not that. It may be one gene per maybe multiple mRNA, which will be translated into multiple proteins. That's how we can make millions of proteins out of only 30,000 genes. Okay, that's very important mechanism. Allows diversity in the number of proteins we have in our cells. That's needed. Okay, evolutionary kind of benefit. So this showed you that intron removal splicing process. So the genome has these, coding and non-coding regions together. When it's transcribed, you make that pre-mRNA first. Okay, so with the leader sequence non-coding region with the polyadenylated tail and everything. And then these introns should be removed, okay? Then exons should be connected. And that's just regular splicing if you go this way, but depending on how it goes, there can be alternative products can be made. That's what alternative product splicing is. Okay, so by having alternative splicing, you can get multiple mRNA made, and from those multiple mRNA, you can make multiple proteins. That's what it says. And they showed you, I guess, various number of introns existing per every single gene. Okay, if you look at these different species. So where's human? So Homo sapiens is right there. Okay, in one single gene, how many introns you have? About 7.5 introns. There are so many non-coding introns within a single gene. Okay, compared to what Saccharomyces cerevisiae, it's what the baker's yeast, the unicellular fungi, which may have less than one intron. Okay, there aren't that many introns there. But fruit fly has about 3 introns per single gene. C. elegans roundworm is about 5 introns per every single gene. And plants, Rhabdops thaliana, about 6 introns per single gene. So yeah, so the more, I guess, complex, advanced the species are, seems like there are more introns existing. That's kind of an interesting thing to observe in the evolutionary trees of the life. Okay, tree of the life. Okay, so if you look at the intron sequence, okay, there are some conservative sequences found within the intron. So if this is exon 3' end, and this is another exon on the 5' end, in between there's an intron stuck in there, right? So if you look at the intron sequence at the beginning, you will be able to find this conservative GA, either AUG, the purine, and then AGU sequence. That's very conserved on the 5' end of the intron. And then in the middle, there's that unique conserved sequence, okay? Pyrimidine, any of the 4 nucleotides, that's what N is. Another pyrimidine, and then U, R, and A, that's very conserved A. And the pyrimidine sequence, that's a branch sequence, and you will always have that A. And then at the end of the intron, you will have pyrimidine and any nucleotide, and CAG is found as very conserved sequence. You will see that intron sequence. Okay. Let's talk about the intron removal, the splicing process. There's a form called lariat formation during the intron splicing. Okay, so what that means is this is a lariat. It's like a lobe, okay, shape. So lariat formation is made. Okay, so that lariat formation is made between the number 2 carbon of the ribose OH in the conjugate A sequence. So that A, okay, it's got number 2 carbon. What it means is it's here, it's that A nucleotide. So that's number 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, right? So that number 2 carbon, so originally that should be OH there, okay? So that is very conjugate A, okay? And that attacks the phosphate of the conjugate G in the 5' area. So we're talking about that guy, okay? 5' G, so which is shown up here in the G here, okay? So the 5' end, okay, is attacked by this 2 OH of this A. That's what it's showing up there. Okay, and then because of that, the lariat is formed. There's a connection. Okay, the covalent bond is formed. Okay, so that's how this G is disconnected from the exon 3' end, and instead it's making the lariat formation. Okay, so the 5' end. Okay, and of the intron is joined to the A through that covalent bond. Okay, so that's the lariat formation. So of course there's an enzyme involved in that. Okay, we'll talk about that. So actually nuclear protein enzymes, okay, ribonuclease enzymes are involved. So that's that. And then the 5' exon, okay, freed from the intron attack. Okay, so here this one intron, and that can attack the phosphate at the 3' splice site. Okay, so we're talking about that exon is attacking that first pair on the 3' end side. Okay, and the 5' end of intron joins the branch side, and we just saw that forming the lariat, right? And then that is attacking that right now. So doing so, you can cut up the intron out and then reconnect the axons together. That's going to be the next step. Okay, so in that splicing process, okay, who's doing that? Now that's the question, right? It's not just done automatically. There are some introns that are done automatically, but the general pathway is there's an enzymatic complex called splicesome that's taking care of this. And the splicesome is made up of 150 proteins, a large complex with about 5 RNA molecules stuck in them. Okay, so we call those RNA, 5 RNAs, small nuclear RNA molecules, snRNAs, okay, and then protein molecules as snRNP, small nuclear ribonuclear proteins, okay, RNA binding proteins. Okay, so snRNPs and snRNAs come together, they are making that huge splicesome complex, and they are doing the intron splicing. Okay, so snRNP, another name for that is SNRPs. So snRNP, those proteins have 3 roles: recognizing 5' splicing site and the branching site, and bringing these sites together to form the lariat. Okay, and then cleavage and joining the reaction, they do that. Okay, and then among those, these RNA molecules U1, U2, U4, U5, U6. Okay, these RNA molecules, snRNAs. Among those, the U1 and U6 RNA, small nuclear RNA, have complementary sequence that can base pair with 5' splicing site in the pre-mRNA. So U1, okay, small nuclear RNA has the complementary sequence that matches up with the 5' end of intron. That's what it's showing up there, right? And then U6, okay, also has a complementary matching sequence with you know, the intron 5' end as well. Okay, that's what it's showing up. That's how they can recognize intron and bind and then catalyze the splicing process. The branch site is recognized by U2, another RNA, snRNA. So it has the matching sequence there again. Okay, so there. But the A, the important A that provides OH, is actually mismatched there, so it's hanging out. So that's the important thing. Okay, see that? U2. No, so that's a branch point of the snRNA. The intron. Okay, so there's a little mismatch there. Okay, and then some non-snRNPs like U2AF and U2 auxiliary factor recognizes the polypyrimidine in the 3' end and helps the branch point binding protein to bind to the branch site, and the branch binding proteins is displaced by U2 snRNP after that. Okay, so we're talking about these guys, BBPs, the branch point, they're binding, okay, U2, so protein binding on that branch site, but A is kind of skipped because there's one nucleotide mismatch. Okay, so the splicesome is important in carrying out the splicing mechanism, of course. So this is a process of splicing mechanism, of course. The 5' end splicing site is recognized by U1 snRNP. There you go. And then U2AF binds the pyrimidine track and 3' end of the intron. So U2, yeah, right there. Okay, and helps it to bind on the branch site. Okay, U2 right there. Okay, so and this makes all the E complex. So that's the E complex. Okay, and the U2AF helps U2 snRNP to bind the branch site. Okay, U2AF. Okay, so here we're talking about that. Okay, to displace BBP. So BBP is displaced. Okay, so that guy displaces the BBP. Okay, and then the base pairing between U2 and branch site makes A nucleotide excluded. So we're talking about that. Okay, and can attack the 5' splicing site. So that exclude A will attack the 5' end. That's what it means.